fermentation presentations by presenting the findings on enzyme activity changes during the fermentations of highland barley yellow wine. So in this study, the objective was to evaluate how different microbial strain ratio influence the fermentation process and product quality. Enzyme activity was a key parameter in assessing the fermentation efficiency. So 3 enzymes were monitored during the study, which is alpha amylase, glucoamylase, and also acid protease enzymes. Starting with alpha amylase, its activity peaked on day 1, particularly in group C and D, but dropped sharply by day 7 across all groups, indicates that the starch breakdowns was completed early in the fermentations. As for glucoamylase, which breaks down starch into glucose, the CK group peaked later around day 7. In contrast, the experimental group, especially group C, show high early stage activity and better enzyme stability over time. Lastly, enzyme protease activity, which contributes to protein hydrolysis and flavor development, declined gradually. However, Group A and C maintain high activity by day 25, suggesting that extended protein breakdown during the later fermentations. So in summary, the enzyme activity results shows that Group C microbial combination supported more stable and effective enzyme functions. This not only enhanced sugar availability and nutrient breakdowns, but also contributed to better fermentation quality, supporting the study goals of optimizing yellow wine production through tailored microbial starter culture.
Hi everyone, let me continue to presenting the result of the study on the physical-chemical properties of the yellow wine. So what is a physical-chemical properties? It refers to the physical and chemical characteristic of a substance that define its behavior and interaction in a different environment. In the context of food and beverage like yellow wine, these properties are critical because they influence how the product quality, taste, and the stability. First, let's look at the reduced sugar level. It was significantly lower in the experimental group. This indicating a better fermentation. For example, Group A had a 3.592 grams per 100 milliliters compared to the control, which is 6.503 grams. Next, the total acidity. It was lower in the experimental group, with Group C at 6.065 grams per liter, the total acidity improving the wine taste profile. The amino acid nitrogen contribute to the flavor complexity. It was lower in the experimental group due to the microbial assimilation. For example, for Group B, it recorded that 0.266 grams per liter compared to the control, which is 0.658 per grams. So in the term of alcohol content, Group B reached the highest level at 15.1%, it showing a superior sugar to ethanol conversion. So finally, for the sensory evaluation score, it was higher for the Group C with a score of 83.1, it reflecting the improve of the flavor and the aroma of the yellow wine. So now, let's moving to the discussion. So the result revealed that the experimental group will show a significantly lower sugar level, this demonstrating the improved sugar utilization. This was primarily due to the microbial symbiont, which enhanced the ethanol conversion and resulted in higher alcohol content, particularly in the Group B. The reduced acidity and optimal amino acid nitrogen level in the experimental group contributed positively to the sensory attributes of the wine. Interestingly, a Group C achieved a high sensory score of 83.1, emphasizing the role of diverse microbial activity in creating a complex flavor. The finding also aligned with the previous research by Peng et al. 2024. It observed that the improved ethanol yields and sensory profiles when mixed the microbial fermentation. Additionally, the lower acidity levels observed correlate with the study and adjusted fermentation kinetics. Finally, for the microbial symbiont, it offers a promising approach for developing high-quality fermented beverage. This ability to create distinct sensory properties could revolutionize the production of specialty wines. So in summary, the microbial symbiont are instrumental in improving the yellow wine's fermentation efficiency, flavor, and overall quality. The application opens exciting possibility for innovation in the beverage industry.
The electronic tank analysis showed clear flavor differences among 5 yellow wine groups using different microbial mixes. The commercial yeast group CK had the strongest sour, sweet, salty, umami, and bitter tastes.
In contrast, the experimental groups A to D were milder, with Group C showing the most balanced and harmonious flavor. Groups A and D had very similar taste profiles, while CK was clearly different from the rest, mainly due to different fermentation conditions and microbial ratios affecting flavor compound production. The results show that different microbial mixtures have a strong effect on yellow wine flavor. The commercial yeast CK produce intense sweetness, bitterness, and umami, likely due to high enzyme activity and flavor compound release. In comparison, the experimental groups A to D had milder and more balanced flavor, with Group C standing out. Its specific ratio of 4112 created the most harmonious taste, showing that microbial synergy and the right strain ratio are key to developing a good flavor through the transformation of organic acid, amino acid, and volatiles. The findings are consistent with previous studies by Xu et al. 2019 and Liskowska et al. 2025 that found increased production of carbon dioxide and biosynthesis of volatile compounds by the mixed culture of lactobacilli and yeast.
For volatile compounds, 77 species can be detected, with Group C having the most species. This is because when Rhizopus is the dominant microbe, it can inhibit the metabolic activities of other microbes, which affects diversity of the volatile compounds, which when compared to previous studies, more than 900 volatile compounds detected, 77 compounds found in this study seems quite low. For the next point, compared to the control group, CK, the experimental group shows higher relative content of volatile compounds. This is because the synergistic fermentation of multiple strains of microbes can increase the content of volatile compound, which can be supported by previous studies by Yang et al. Therefore, even though having one dominant microbes affect the diversity, the use of multiple strains can increase the content of volatile compounds. Among the compounds detected, esters is the most abundant, with hexadecanoic acid ethyl ester is the most abundant compared to the ester group detected. The high diversity of esters is due to the production of ester using its corresponding alcohol. And with the hexadecanoic acid ethyl ester being the dominant compound, it may be the significant reason behind the flavor of the wine. And finally, the control group CK having more phenylethanol content. This may be due to some type of yeast producing phenylpyruvate decarboxylase, which increases the amount of phenylethanol production.
Yellow wine is often referred to as a liquid cake because of its high nutritional value, especially its rich amino acid content. During fermentation, amino acids are released primarily through two processes: proteolytic activity from microorganisms and yeast autolysis, which break down cell components. In this study, a total of 17 amino acids were detected across all sample groups, including the control and the mixed strain fermentations. Among these, glutamate or GLU show the highest concentration in every group. This is important because glutamate contributes strongly to umami taste and also act as a precursor for key flavor compounds such as ammonia and tetramethylpyrazine. Other major amino acids present in high levels include aspartate, alanine, leucine, arginine, and proline. A heat map from the study clearly shows that glutamate was significantly elevated across all treatments, reinforcing its central role in sensory profile of highland barley yellow wine. Amino acids were also categorized based on their sensory properties. Umami types like aspartate and glutamate, sweet ones like alanine or glycine, bitter ones like leucine, isoleucine, and lysine, and salty amino acids such as cysteine and methionine. Interestingly, the cold fermentation treatments slightly reduced the level of bitter amino acids, particularly phenylalanine, leucine, isoleucine, and lysine. This is also due to the increase of activity of Ehrlich pathway, which converts amino acid into flavor-active volatiles like higher alcohol. Overall, this shows that by adjusting the fermentation strengths, we can actually influence the amino acids composition and in turn fine-tune the flavor and quality of yellow wine.
The study found that using a mixed culture of these organisms at 4:1:1:2 ratio improved the taste and quality of highland barley yellow wine. The wine had a balanced sweet-sour flavor, moderate sugar, good acidity, and 13.1% alcohol. The mixed strains also maintained enzyme activity during late fermentation, boosting the production of volatile compounds, especially hexadecanoic acid ethyl esters, which likely enhances the aroma.
All wines contain 17 amino acids, with glutamic acid being the most abundant. This shows that adjusting the strain ratios can enhance both flavor and aroma through synergistic fermentation. That is all from our group. Thank you.